We're going to talk about meiosis. In meiosis, a diploid cell undergoes two cell divisions, producing four haploid cells. The orange ring represents the cell wall, the orange boxes represent the centrioles, the green circle represents the nucleus, the yellow ring around the nucleus represents the nuclear envelope, and the blue lines represent the chromatids. This cell is undergoing interphase. During interphase, DNA is replicated. This cell is undergoing prophase 1. The pink X's are maternal homologous chromosomes, and the purple X's are paternal homologous chromosomes. The pink lines are the developing meiotic spindle, and the yellow dots are the kinetochores. During prophase 1, the homologous chromosomes lie lengthwise, side by side, through the process of synapsis. The homologous chromatids exchange segments of DNA strands through crossing over, which occurs in the latter part of this stage. This cell is undergoing metaphase 1. The pink lines are the microtubules, and they are attached to kinetochores. The centrioles move to the opposite side. During metaphase 1, tetrads are held together by chiasma. They line up on the midplane. Both sister kinetochores of one chromosome are attached by spindle fibers to the same pole. The other sister kinetochores are attached to the opposite pole. This cell is undergoing anaphase 1. Each pair of homologous chromosomes separate and move toward opposite poles. Each pole receives a random combination of maternal and paternal chromosomes, but one member of each homologous pair is at each pole. Sister chromosomes remain attached at their centromeres. This cell is undergoing telophase 1. During telophase 1, cytokinesis occurs. Each nucleus contains the haploid number of chromosomes, but the chromosomes are duplicated. These cells are undergoing prophase 2. During prophase 2, a brief period of rest occurs called interkinesis. Centrioles duplicate, the nuclear envelope breaks down, and the spindle apparatus forms. DNA does not replicate. These cells are undergoing metaphase 2. The chromosomes line up on the midplanes of their cells. For each chromosome, the kinetochores of the sister chromatids face the opposite poles, and each is attached to a kinetochore microtubule coming from that pole. These cells are undergoing anaphase 2. During anaphase 2, the chromatids and the centromeres separate. The chromatids move to opposite poles. The separated chromatids are then called chromosomes. During telophase 2, there is one representative for each homologous pair at each pole. Nuclear envelopes reform, cytokinesis occurs, and 4 cells are produced.